from NABI, that is National Agri-Food Biotechnology Institute, Mohali. Further, for his postdoctoral training, currently he is working as a postdoctoral scientist in University of North Carolina, Chapel Hill. Dr. Jain is working in the field of plant biology, especially plant abiotic stress. And today, Dr. Jain will talk about pushing boundaries, how external stimuli affect meiotic recombination in plants. He has published several papers in this field and also received several awards. So I invite Dr. Pratik to come on the stage and talk about your lecture. Thank you.
Thanks, Naveen, for the kind introduction, and thank you, everybody. Not because I come here, but for your patience since last 20 minutes. And I accept my apology, my fault, right? Anyways, before going further and to my talk, I just want to go in the old Indian history, especially about the movies. If you remember, I hope everyone knows about the Mughal-e-Azam, right? So when the Mughal-e-Azam was released, it was in the black and white, right? But few years ago, during the Diwali, it came in the color, and everyone was amazed, right? Even my mom. So she usually not goes to the movie theaters to watch the movie, but for that movie, with my aunt, she went to the mall. First day, first show, 12 p.m. 12 p.m. Right? And why I just put that reference over here? Because even the people who are fan of the black and white movies or the colors, so when they see the colors, they feel amazed. So can you turn off the light right here? Okay. So even they see the colors, they feel amazed, right? And when we talk about the things which are going inside the biology or in the living cells, so no one can counter the amazement of the colors, right? So if you see here, so by using these colors, I was trying just to understand the effect of the external stimuli on the meiotic recombinations. And why I just put this title for my presentation, the pushing boundaries, because usually when we say the recombination, it's mostly the natural, right? So there's some natural effects, some nature gift, right? So that pushes us, or means that pushes the cells to go for the recombinations, right? But can we control it or not? So that's the whole theme of my presentations, right? So before going further, can any one of you, just by looking these pictures, can tell me what this can be? This picture. Wait. So these are the pollens, right? The pollens, not the black and white, but the color one, which are expressing the three different proteins: the red fluorescent protein, yellow fluorescent protein, and the cyan fluorescent protein. And by using colors, I did my whole studies to understand the effect of environment on the meiotic recombination. Right? To go further, when we say the meiotic recombination, the meiosis is very well defined. I know some are very young faces and some are the old one, like our director, sir. Right, so who has very well idea about the meiosis. But to go in the fundamentals, I just want to brief. So when we talk about the cell division, there are the two different types of the cell division. One that usually happens in your somatic cells, which we usually call as the mitosis. And the other one, which usually happens in your germline cells, that I am considering, or we all know as a meiosis. Right? And there's one more fundamental difference between the mitosis and the meiosis, is that, so when we go to the M phase, that we usually call divided into the 4 different stages, or maybe the 5, the prophase, metaphase, anaphase, and the telophase. In the meiosis, there are 2 different M phases, which we usually called as a prophase 1, metaphase 1, anaphase 1, telophase, and subsequently for the M2. But when I can go with the recombination, and we go with the recombination, it usually specific with the prophase 1. And if I goes with the prophase 1, which is basically subcategorized into the 4 different classes. Just a minute. Yeah. So 4 different classes. One is the lactotin, the other is the zygotin, okay, the other one is the pectin, and the other is the diplotin. So definitely all stages are important, and not only the prophase one, but all those which goes before M phase and after M phase. But if I focus in the lactotin stage, it is the stage where the DNA starts to condense, right? So they align themselves for the next stages. If I go with, then it goes with the zygote. In the zygote, basically, one more thing is that, so during the lactotin, there is a breakage of the double-strand breakage occurs with the specific help of the specific kind of protein, that is SPO11, that chop of the DNA randomly at n number of the sites, right? So, but in the case of the zygote, now the question is that, so if there is a different strands of the DNA, how those strands basically participate in the mitotic recombination? So they cannot go by themselves and just attack or invade on their counterparts or in their sister chromates. Yes, now that definitely they are the role of some other cofactors. So basically that help those chopped DNA to invade their sister chromates. And those DNA, so there this whole process is over there. First of all, as there is a chopping, we all know there is just trimming occurs, and the trimming at the 3' OH end. So which basically release one free ends of the DNA, and in the zygote stage, so this strand invasion basically occurs. And if you see here, there is a crossing over is basically started. In the later stage, the pectitin, this crossing over is going to finish, and both of the sister chromates, if there is a perfect crossover is happen, so now they are sharing their parental sister chromatid strands, and which is basically end at the diplotin stage. So if you see here, there are the several different factors are involved during this process, not only the SPO11 protein, but also either ZIP5 and other different classes of the protein. So as I mentioned earlier, but I'm just more focused in this phase where actually the strand invasion or the recombination is happening. So the genomic region is basically participating during the crossover. So, but if I'm saying that there are the different sites basically which are break during the recombinations, but is all is participating in the successful crossover or not? So on the basis of it, there are the two different pathways has been given. So once if you see here, if there's a breakage occur and the chopping off occur, so there the involvement of the two different proteins which basically control whole this invasion. And on the basis of it, we have categorized into two different paths. One is basically the type 1 pathway, which is basically regulating around 85% of the recombinations. And one is the type 2 pathway, which is regulating around 15% of the recombinations. And X of it, so there are some other like products we can say, which are basically considered as non-crossovers. What do you mean by the non-crossover? That means the gene conversion, means that strand transfer is only occurred in the one of the sister chromates, not both. So these are the basic models which are defined for the mitotic recombination. Now again the question comes, okay, so there are the n number of the questions, right? So as we see, the people, there the whole war is going on, there the environmental calamities here. Why you are not studying on this? You can go, you can study. But for me, why I decided to interest in the crossing over, first of all, I'm a plant scientist. Since last one decade, I am working with the plant, so there is my affection. And second thing, as about if we go with the India, so we India is we usually say we are agriculture prominent country. I know there's a lot of boom is going on in the IT industry, but there's a lot of farmers who are basically depend on the agriculture, right? So this basically excites me, and it also like pushes me and identify one of the problem is in the front of the breeders is about that. Not if there's a paper published and they talk about that if there's a whole DNA is available, why not whole DNA is participate in the mitotic recombination? Means it can be. So there the two pathways have defined, and the people have just sequenced the genome of the one of the model plant Arabidopsis, and also with the cereals, they found that in the Arabidopsis, the most of the recombination is basically occur at the telomere end. Although you can see there is a couple of the sites which are basically, if in the red, you can see there are a couple of the regions which have the higher crossover. But if you compare it with the cereals, the crossover is usually happens only at the ends, at the telomere, not in the midfield. That means the main part of the whole genome is basically recombination poor. That means they are not participating in the recombination. That means if there is a gene that that you can understand it like this way. So if this is like they are the two ages, right? So they are just participating in the recombination. And this internal region is responsible for the higher yield, right? And there is no yield, but it gives you a higher height, right? But both of the regions are comprised into only the centromere part. But when you cross them together, they are not basically merging with together. Means whatever the progenies you will get or the seeds you will get, they have only just one phenotype, not the other. So the question again comes, so can you push the boundaries and can you push these intergenic regions, or I can say the intergenic regions for the recombinations or not? Then I ask the second questions. Okay, let's suppose you push it. Now, as being a scientist, what is the molecular mechanism? So that's always a question. So if you know something, then you should identify the reason behind it, right? And the third one is the repetition of the first questions is to induce the hyperrecombination. So if you see here, so this is one of the experimental line, or you can say the wild line. So let's suppose if I consider this yellow mark as a disease resistance, right? But with this disease resistance, there is a one trait is also attached, which gives you the small height. And there is this elite line, which gives you a higher yield, and there is a big height, but it is not disease resistant, it is a disease prone. So if you cross them together, right, it goes with a normal recombination due to the linkage drag, these traits basically goes together. That means you will get a line which gives you a disease resistance, but it will be in the small height, right? But if you somehow you break the interactions or the genomic region between these two traits, you will get the line which has only the disease resistant phenotype, and it is in the elite line. That means by breaking the linkage drag, you will have the lines which are disease resistant, and it will give you a higher yield. And that's a boon for the farmers and as well as the breeders. So, so that means, and how it can be done? It can be done by creating a roasted. So we all know, so in the freezer, so there is a temperature roasted. So you can just play around, I just want the hyper cool condition, right? So you just turn on the switch, just move the roasted. So why not for the plant system? So I just want to develop that roasted. Okay, I just want to induce the mitotic recombinations, right? So I spray something, or I just put the condition, and there's high recombination frequency, and don't want to depend on the nature, right? So in that case, so why it is beneficial for the recombination, that it basically overcome the linkage drag, it support the elevate the genetic diversity, it increase the crop yield, and it is novel genes for the disease resistance. So when we say, now the question is how, right? So when we say the recombination is usually happens in the germline cells, right? So if I consider as a plant, in the plants there are the two systems where the recombination is happening. It usually happens in the female part or in the male part. So in the female part, I go first, it usually happens in the egg cells, eggs which are which resides in the ovary, right? And in the just male part, which usually happens in the pollens, and which present inside the anthers. So for my study, I use the pollens as a tool to understand the recombinations, right? So if you know, the pollens are also the product of the meiosis. So so there's a whole pathway, is very well defined pathway that is considered divided into the microsporogenesis and the microgametogenesis, where inside the anther there is a pollen mother cells usually present, and those pollen mother cells undergoes the meiosis that will result in the pollen formations, right? So by studying so why what are the advantages of these? The advantage of these is the ample sample that is not possible with the female part. It is easy to isolate, means you just tap the flower, then you will have the n number of the pollens, right? And it is effective and it is a time efficient, and that is not possible for the female ones. So the question is that how we can use those pollens as a tool. So usually if you see the pollens, so they usually, if you want to isolate the pollens, means you can do these experiments in your lab, like anyone, like in the undergrads who are in the first year or the second year. Just take a flower, tap on the cover slide, any flower, not the Arabidopsis, right? You will see the yellow grains, right? So, and those yellow grains are the pollens, not the big one. Means if you go with the rice, you will see the lot of anthers. But in the small flowers, you will see the yellow grains, and those are the pollens. But the issue with those pollens, that they usually present in the single one. And it is not possible to see the recombination. Means you can see the recombinations, but it is not very time efficient. So for the time efficient, so like 2 decades ago, a mutant was identified, that is a quartet mutant. And if you see in this image, so these are the 4 pollen grains which are attached together. Because there's one of the gene that is a QRT1, that's a quartet. It is basically mutated in this gene. So after the post-meiosis, all those 4 pollens are not able to detach from each other, and they are attached together. Right. So what we did, we basically fused like 3 reporter proteins. One is the red fluorescent protein, one is the cyan fluorescent protein, and one is the yellow fluorescent protein under the post-meiotic promoter, that is the LAT52, and just express in the plants. So how? So first we made the plants which are expressing only the red fluorescent protein. And then we made the plants which is expressing only the cyan fluorescent protein, right? So the question is that, so now, okay, so these are the 2 plants, right? So one is expressing the red fluorescent protein, and one is only the cyan fluorescent protein. I want a plant which is expressing both together. Yes, you can use some genetic tools or biotechnology tools, you express both these proteins. But what we basically crossed these 2 plants together. And after n number of the screening, we found a plant, or I can say the line, which is expressing both these proteins. But the ratio is the 2:2. That means 2 of these pollen grains are expressing one color, and 2 of these pollen grains are expressing the another color. We did the same with our third the fluorescent protein, that is a yellow fluorescent protein. In the yellow fluorescent protein, we did the same thing, and we found a line which is expressing the 2 pollen grains as the yellow fluorescent protein and 2 as a cyan fluorescent protein. But just to make a novel tool, what we did, we just crossed all these together. And after n number of the screening, we found a line which is basically expressing all these protein markers. But there's like a thing here. So in all these proteins, or all these fluorescent proteins are present on the same chromosome. Not like this, that one is expressing on the one chromosome, one is expressing on the other chromosome. Right. So that's why how we can use these systems to study the meiotic recombinations. And these lines are basically labeled as a fluorescent tag lines. So any of you are like can interpret interrupt me in between the presentation, right? If you feel any issues or have any confusion. Okay. I'm going too fast? No? Okay, it's fine. Anyways, now I can imagine myself over there. It's fine, right? So this is the basic mapping over there, right? So how we came to know that the insertion is basically on the one chromosome, not the other chromosome, right? So we use the advanced sequencing method. We sequence whole plants, right? Whole like DNA of the Arabidopsis. And after the sequencing, we found that some of the lines have the insertion only in the one chromosome, right? Some of the lines have the insertion on the second chromosome, some has on the third chromosome, some has 4, some has 5, right? Why only 5? Why not 6? Because Arabidopsis has only 5 chromosomes, right? So that's why we cannot go up to 6 chromosomes, right? Okay. Anyways, so later on. If you see over here, so you can see one over here as a red, right? So we say the gene over here is the red fluorescent protein, right? If you see over here as a blue, so when we sequence it, we found there's a gene over here as a cyan fluorescent protein, and same here as a yellow. So we see the gene over here is the yellow fluorescent protein. And so you see over here as a small like green line over here. Just imagine as a green line as a DNA strand, right? So DNA strand, what does it mean? DNA strand which has lot of bases. There's a whole sequence A, T, C, G, right? Not the U. Why? Because it's present in the RNA, right? So that's why, so you can say one red, then a sequence of the red, that is A, T, C, G, C, G, C, G, T, A, T, T, something, 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 and dot, 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 right? So there's a whole like DNA region over here. And the region over here we called as an interval, right? That's an English term, nothing like a hi-fi sense over there, right? The interval. So if the gene region between the red and the blue is as called as an interval, right? And because I found it is, if I'm taking an example, it is present on the chromosome 1, so it considered as an interval I1, right? And if I take, means in the like coming slides, you will see I use lot of terms like the I2, I3, I5, right? So you can go, oh, there's an insertion at the second number of the chromosome, right? And the second chromosome. If I say the I5, there's an insertion of these reporter markers on the 5th chromosome, right? And you see over here, like A, B, C, D, right? So there is no such signs over here. So that's why, how we sequence it. So when we sequence the reason, we found, because right nowadays it's very, like we have a like longer read sequencing, like the nanopore sequencing or the PacBio or something. But a decade ago, there was no such type of the sequencing, right? There was shotgun sequencing. So whatever the sequences we found, we just categorized them. Okay, we found it first, then we labeled as A. We found it second, we labeled as a B, right? So when, after this, we labeled this randomly I5A or I5B. And those lines we are using right now, and we didn't change it, right? So that's a whole thing about this, how we develop the FTL lines and how we basically name those FTL lines, right? Are you fine? Okay, no sound. Means everything is going above the head, right? No? Okay, anyways. So I'm just coming on the like main theme of my work, like about like pushing boundaries and how external affect mitochondrial combinations, right? So just imagine, I just want to go in the evolution, right? I don't want to argue with some like orthodox like Krishnists or the Hindus or the Jews or something like that. I just want to say, being a scientist, so if we go with the evolution, there were some bacteria, like just go before the bacteria, there are some chemicals. Due to some XYZ chemical reaction, so there are some novel compounds, so the proteins comes into existence, right? They will go further, we got, I don't know, we got or not, but go with the theory, like we have some DNA, and that will enclose in some like organelles kind of structures that will, we have the endosymbiotic theory that will further grow. We have the bacteria, Again, bacteria then goes to the prokaryotes, to the eukaryotes, and the fishes, amphibians, mammals, and non-chordates, chordates, eukaryotes, and as we, right, the human beings. So if you think why these cells, like small like bacteria, evolve in the human beings or the humans or like multicellular organisms, maybe they are smart, right? I don't know whether bacteria, because no one has reported that bacteria has a mind. So they are not smart, right? So, and the other factor is their environmental condition. That means environmental condition somehow affecting the recombinations. Although they were haploid, but somehow there are some changes in their genomic sequences, and they have secreted some novel proteins. And they pushes those haploid prokaryotes into the like diploids, eukaryotes, and goes on, right? So, we ask this question, so giving in the given conditions, as we know the global warming, the temperature is rising. So can temperature is basically affecting the mitochondrial combination or not, right? So as a plant scientist, so we usually use Arabidopsis. I don't know how many you are aware as Arabidopsis. If you means you can just Google it, right? It's a basic weed we usually use to study for the basic sciences, as like animal scientists use the mice, right? So this is the like typical picture of the Arabidopsis. So so in our theory, if we like give the heat stress to the Arabidopsis, not very high, right, a moderate heat stress, so we hypothesize there might be a chance of increase in the crossover rate or in the recombination frequency, right? So to go is further, we again use this FTL system. So if you consider, so in the meiosis, right, if you goes and just remind all those fundamentals, there are the 4 strands, right, 2 from the one parent and 2 strands from the one parent, which are usually called as the sister chromatids, right? So if I say like one of the strand has insertion of like one reporter, and other of the homologous proteins have expressing the different like fluorescent protein, right? So 2 conditions occur. Either they participate in the recombinations or either there is no recombinations, right? So if there is no recombination means they are not exchange of the chromosome, and most of the time we do not see the recombinations, right? You will see the phenotype like the parent type, right? Means 2 as a red and 2 as a blue. But if they participate in the recombinations, means there's exchange of the strands. So you see here, like here means only the one strand is participating. And you will get over here 2 as a parent type, but 2, these are the recombinant type, right? That means by using FTL lens, we can see that recombination is going on or not, right? And so this method, so this is very high throughput, means you can tap the pollens on the slide, you can see the slides under the microscope, and you see the colors. If there's a color differences over there, then you can like use the advanced statistics, whether there's a recombination is happening or not, right? And but this looks very simple, just see under the microscope. But is it really it is simple? Because it is not. So as a population genetics, so we always consider, there are a lot of data is coming nowadays about the India, right? Like about the sex ratio, about the education ratio, and some are basically arguing that Indian population is very big, it's more than 1 billion. And if you are only considering on a few thousand, it is not possible to justify the actual trend, right, about everything. So same study is about these kind of genetic assays, right? So for that, you need the n number of the pollens. Means I score more than 1 million pollens to like prove my theory, right? But every these type of the calculation have a strong background of the mathematics or the statistics. So we have used with one of the professors, I just forget in my mind the name of the professor, but she's a mathematician, she developed these kind of the protocols for us. And it is well defined from the last few decades. So this is usually called the Perkins mapping equations, and this is the novel, the formula behind it. I just don't want to go much deeper, but I just want to say like few terms, introduce few terms over here. One is this PD. It is a parental ditype. That means the pollens not participating in the recombinations, right? One is the tetrad type. That means only the products you will get 2 as a parental type and 2 as a non-parental type, means as a recombinant type. And as an NPD type, that means all those 4 strands which are involved in the meiosis participate in the meiotic recombination. So after scoring all these things, we want to see whether they are basically statistical proof or not. So we basically do like map them, and we use some statistical like the variance. We want to see whether we have calculated the z error, like z score, whether those errors are basically significant, and at last the p-value. So that's like a good and a bad thing about the biology. We all say we can see through eyes, means means I can see the difference, but until unless we don't have the number, we cannot prove our theory, right? And those theory is defined in the terms of the p-value. That's how the level of the significance, right? And we in the so that's how the biological system is different from the other mathematical system or the engineering system, because the things inside the biology are not, they are reproducible, but through the trend, not exactly identical, right? So for that, so we basically consider the p-value. And p-value we usually consider at the 0.05, right? So that means we allow up to 5% of the error, right? So all of these mathematical calculations we have used to prove our theory, whether the effect of the temperature is on the mimetic recombinations or not, okay? Okay, so as I told earlier, so we cannot give the very high temperature, like 45 degrees Celsius. I'm from Rajasthan. In summers, it is, the temperature is around 48 degrees Celsius, right? So if I grow Arabidopsis over here, it will die, right? So we first want to see, means what extreme temperature we can go. And we found that if you grow the plants at 28 degrees Celsius, and if you use these FTLs, you will see the higher recombination compared to the 22 degrees Celsius. That means by putting the plants at 20, or not all the plants, but only the Arabidopsis, at 28 degrees Celsius, you will see the higher recombination frequency. And for that, and this effect is not basically spatial, that means not localized or the specific of the one chromosome region. It is global, means you can see, if you see the I3, that the insertion of those protein marker at the third chromosome, or as well as the fifth I5, you will see the higher in the recombination frequency, right? So, okay, now we know, okay, so that there is a higher recombination frequency, right? But the question is that how this is happening, that's the other thing. So, I just wanted to say it again. So, I show that 85% of this recombination is regulated by the type 1 pathway or class 1. And the remaining is the class 2, right? So we want to know, like, class 1 basically involved in this mitotic recombination or the class 2, right? So we develop some of them. So this class 1 pathway is basically regulating n number of the proteins, like not the n, but a few proteins, which are categorized under the ZMM proteins, right? So in the ZMM, one is the MUSH4, which is one of the key protein and regulating this recombination. And the class 2 crossover is basically supported by the MUS81 or the FANCAM. So what we did, we again just developed the FTLs under the mutant backgrounds of the MUSH4 and the MUS81. And I again want to repeat those things. If there is a recombination is happening, then you will see, if the effect is happening on those mutants, you will see the higher recombination. If the effect is not happening, they are resistant or they are involved in this recombination, you will see no recombinations, right? So we again did all these genetic assays, and we found that MUSH4, if you see here, the control have a higher recombination, but in the MUSH4, there is no recombination. And this trend is followed for both, for the I3A as well as for the I5C. Yeah.
Sorry to disturb. What about flowering of these mutants?
No, so flowering, like for the MUSH4, it's there was a flowering is fine, but
No, with temperature, are they flowering early?
yeah, it's fine at 28 degrees Celsius.
Are they flowering
Yeah,
early?
yeah. No, well, no. There's a protocol just to this kind of treatment. It's not like we are putting those plants in the early stage. So we put the plants when they are flowering, right? So because the pollens usually present inside the flower, unclosed flower, right? So when we know that so we know the flower buds are coming, but they are not too old, then we put those plants under the heat conditions. So after 5 days, we will see the flowers over there, and then we isolate the pollens, right? Anyways. So we repeated this experiment with the MUSH4 as well as the MUS81, and we found the MUSH4s are resistant, means we will not see any recombinations. That means it shows that the MUSH4 basically involved in the meiosis, right? But when we give the same heat treatment to the MUS81 mutant, we saw the higher recombinations. That means this is not involved in the meiotic recombinations. That only the MUSH4 is basically involved. And the next question is that, so we did some cytogenetic experiments, and after it we found that if you see the MUSH, there is no significant difference there's a like in the Musk 81, which treated at a 28 degrees Celsius, there's a higher recombination as like the wild type. For that, we use some recombination specific antibodies, that is a MLH1. Yeah. So as I said earlier, why not the higher temperature? Right. So there are a lot of studies that are come, like especially if I go with the Arabidopsis, and they compare, like if you go with the 18 degrees Celsius or 20 degrees Celsius, and if you grow Arabidopsis under high heat, up to 32 degrees Celsius, there's a defects in the pollen formation or in the development. And not only with the Arabidopsis, the issue is also with the wheat and also in the poplar. That means we cannot generalize this treatment for all crops, right? And in the natural condition, it is not possible. So what are the other modes which can be helpful for these recombinations, right? So then we ask another question. So there are a lot of studies are over there. And if you see here, sometimes we say that if you eat this fruit is basically for the cold, like our dry fruits, right? That means they have some properties to generate the heat. Same is with the chemicals, right? Some of the chemicals which are basically similar to the heat, some of the chemicals are similar to the cold, and some are the pesticides, and maybe they have some effects on the plants. So we have categorized and made a whole list of the chemicals, like about 200 chemicals or maybe 250, and which belongs to the different categories. They belong to some food additives, they are from the pesticides, they are from the insecticides, they are from the fertilizers, and they are from the stress. And what we did, we just we make the vials, we spray those chemicals on the plants, especially when they are undergoing the flowering, right? So not very early, because it will take around, after germination, it will take around 4 weeks for Arabidopsis to flower, right? So, and second thing is that, because the pollen usually develop in the flower, right? So we are more focused on the flowers. So, and what is the advantage of this chemical accelerated mitotic recombination? So first of all, as I discussed earlier, it might be helpful to break the linkage drag, and it might be improved to improve the poor agronomic preferences. It will be helpful to generate to improve the phenotypic challenges. Recombinations in the cold genomic regions or like poor recombinations regions. It might be helpful to remove the labor-intensive like crossing or the hybridization methods. It will be helpful to generate the superior cultivars, and it will be helpful to lower the inbreeding depressions. So these are the like measure of the advantage we basically focus to apply these chemicals for the treatment. So as I mentioned earlier, I screen approximately 150 chemicals, and I'm saying more than 200, and that's more than 200, right? Anyway, so I'm not showing the details of those chemicals, due to some proprietary. My apologies for that, and due to some patent-related issues, but we categorize these chemicals in different codes. Like, as I'm discussing more about the heat stress or the temperature, so HS4, HS5, HS7, HS8, HS14, 15, 20, 24, and 26, right? So, and when we did the same studies again, and we did some genetic assay. When we treat those chemicals, when we treat the plants with these chemicals, we found there's a higher recombination as like the temperature stress. Means the mode of action of these chemicals is similar to the temperature stress. Same with all chemicals, right? HS5, HS7, HS8. And for this, we use the 5 millimolar concentration, 12.5, 15 millimolar, 2.5, 2.5, and 0.5. Now, again, the question comes, why only this concentration, not the other? Why not? So the answer is that before going these concentration, I did some dose-dependent assay from like 0 millimolar up to the 10 millimolar, maybe 20 millimolar. right? So, and after that we found that the most of the chemicals are basically showing the bell-shaped curve. Means one of the concentration is basically has a maximum effect, not the edges one, right? So again, so the question is that, so we know that these chemicals is basically affecting the mitotic regroupation. Now again the question comes about the molecular mechanism. Are these chemicals are following the same pathway as the temperature stress or not? Right, so we again just tested these chemicals on the MUSH4 and the MUS81 mutants, and all these chemicals have the similar effect as like the temperature stress. The chemicals are basically not affecting on the MUSH4 in both genetic backgrounds, I3 and I5C, but they have a higher effect on the MUS81, right? That means the chemicals are basically following the same pathway as like the heat stress, that is a type 1. So, but these things, I'm just showing only the effect of the one chemical due to this time limitation, right? So, anyways, so now the question comes, okay, so you did a treatment, right? You have some effect, but whatever the effects you are showing is at the genomic level, means at the DNA level, right? But there might be some pathways, right, which are basically involved or which are working as a carrier for taking the effect of those chemicals from the surface of the plant leaf, right, to at the core, inside the nucleus, on the DNA. If we divide whole this pathway, there are the n number of the proteins that are involved, or there's the n number of the receptor. It is very hard to pinpoint, right? So I did some like survey, I did some literature survey, and I found like a decade ago, there's a protein was involved. Yeah, but before that, so as I told you, so there's some reason basically which are the recombination poor reason, right? So I say the recombination poor reason, so if somehow you like modulate those recombination poor reason as a recombination fair reason, that means you will somehow increase the recombinations, right? So I targeted the SWI SNF remodeling complex, if you just remodel it, right? So there's a reports which talk about the plant sensing, how basically the plants sense the temperature, right? And they reported about one of the protein, this is the ARP6. So this ARP6 basically a part of the SWI SNF remodeling complex, and if you remove this protein, the plant stops to sense the temperature. So I asked my question, is this protein is involved, if this involved in the temperature sensing, somehow it is also involved in the mitotic recombinations, because this is the key protein for this sensing. So I again developed some mutants or the FTLs of this ARP6, and yes, the mutant of the ARP6 are non-responsive to the temperature stress in the both genetic backgrounds, I3A as well as the I5CD. And for that, but the question is that, so is the chemical treatment also following the same pathway or not? So I used one of the chemical, HS4, and did the same treatment on the flowers, on the plants, and found, yes, the chemicals are also following the same pathway as like the temperature sense. That means chemical effect is not responsive on the MUSH4 mutant, but it is responsive on the MUS81. That means the chemical treatment, that means, I'm sorry, not the MUSH4, MUS81, but the ARP6. That means the role of the ARP6 is common for either for the temperature stress or either with the chemical treatment. So now the third one. So first I told you how the breakage, one of the two key proteins, MUSH4 and the MUS81, which involve in the strand transfer. The second I talked to you about the epigenetic modification, which is involved the ARP6. But again, the things come. What is going into the cytosol? Because those two, ARP6 or MUS3, either ARP6, either MUSH4, or either MUS81, they are basically present inside the nucleus, not in the cytosol. So they may be some of the key transcription factors or other things. So I focus one of the key transcription factor, that is the HSFA2, that is the heat stress factor A2. Why HSFA2? Because it is one of the key transcription factor first defined, and it is highly expressed during the heat stress. So I just want to give more focus on the HSFA2. It is the strongest expressed member, and The other thing is that, which is also important, that it is bind with the estrogen lysine 4 hypermethylation, and that is basically the site for the ARP6. So that's how all these three are related. The HSFA2 bind to the this estrogen lysine 4, which is also the binding site of the ARP6. So this HSFA2 basically work as a trimer, means three of the HSFA2 they combine together, and they binds to their target sites for the expression of the genes, right? So to identify the role of this HSFA2 during the mitotic recombination, what we did, we again developed the FTLs for this in the HSFA2 background, and the result were fantastic. Those HSFA2 mutants are basically non-responsive to the temperature stress. And also when we tested, I'm sorry. It's got stuck. No? Okay, thank you. Yep. So not only with the temperature, we also did some chemical treatment, and we found the effect is similar. The HSFA2 mutant is basically non-responsive to the chemical treatment. So on all of these experiments just to pushes us to make the hypothesis, and if we see here, the higher temperature has is involved. If you give the higher temperature, not very high, but 28 degrees Celsius, you will see the higher recombination. It basically induces the ARP6, HSFA2, and it will give the higher recombinations. But the chemicals, especially the heat mimicking chemicals, are basically working similarly like the temperature stress. And if you spray the plants, we don't know about other interacting partners which are involved on this chemical sensing, and how these chemicals basically going towards to the DNA. But we know that role of some of the key proteins or the transcription factor, like the HSFA2 or the ARP6 or the MUSH4 and the MUS81 in the mitotic recombination. So I have 10 more minutes. Okay. So yeah, so these are the like 3-1. Now the question is that anyone can ask, well, in the biology there are the n number of the factors. Why not others? Right. So maybe these are the not key. These are such substitutory. Right. So we should ask our questions, and we should like do more research just to identify other factors. So for that, so what I did, I EMS mutagenize, and I'm assuming many of the people know, and some I don't know. But EMS is the ethyl methyl sulfonate chemical, so that if you randomly mutagenize, that generate the mutations at the random positions inside the DNA, on the DNA. And that might be rescued some phenotype, or maybe it is responsible for the loss of the function. So we just want to know, so that concentration I use over here is very high. It is means for the HS4 treatment, I use 7.5 millimolar for the treatment. And why 7.5 millimolar? Because we are just spraying, and when you spray something, it is not only the 1:1. Some are basically going inside the cells, and some are basically going into the environment. So we don't know. So for that, so first we want to know what is the toxicity level of this HS4. So we did some dose dependent assay, and we found like up to a 300 micromolar concentration, there is no germination. Means all the plants will die, right? So we did some mutagenize and with some mutations, and we found some of the lines which are basically growing at the 300 micromolar. And means, I don't know, like maybe there's mutations at some receptors, or maybe some of the proteins which are basically involved in this uptake, or maybe in some other, have some functions we don't know. So, but we found some of the lines which is still undergoing, and we are just making the, we are going to just genome sequence just to find out other genes which are involved in this chemical sensing. Now the third question is that which is also undergoing, like can we, because now there is no such a temperature barrier over here, so it's just a spray treatment. It's very easy to use. So can those chemicals effects on the monocots, like on the rice or maybe on the wheat or others? So I developed some hybrids. Now the question is that why you need the hybrids, right? Why you need the F1s, why not the parents? Because if you take the parents, because all parents, if they are the homozygous, right, so you will not see any changes in the recombination, because all four genomic regions are the identical copies of each other, right? So for that you need the F1s. So I made some F1s, I did the chemical treatment, and because I just told you earlier, it is not possible, maybe possible, but it is very hard to do this for the female one. And for the rice, we don't have this FTL type of the system to understand the effect of those chemicals on the recombination. So I isolate the anthers and later on the pollens, and send them and isolate the DNA from these pollens and send it for the long-read sequencing using the nanopore. I have the data, but it's still analyzing, so I'm sorry, I'm not able to discuss it here, right? So, but anyway, so this is the like third goal of the chemical treatment. So at the end, I just want to summarize all those things, that chemicals mimicking heat resistance could be an alternative tool to accelerate the mitotic recombination in the plants. Temperature and the heat mimicking chemicals share the similar pathways to stimulate the mitotic recombination frequency. And our studies has established the novel use of the heat mimicking chemicals for the mitotic recombinations. We showed the chemical induced mitotic recombinations follows the type 1 pathway, and also involvement of the other key proteins, that the ARP6 and the HSF82. So, do we have a time? Okay. Okay. So, I just want to like, so this is the main projects I am still undergoing. But before that, I did I work on two exciting projects. I just want to share a glimpse, not all. Like, and one is about development of the seedlessness. Like, can you get the seedless berries? Can you get the seedless tomatoes, right, or not? If yes, then how? So during my PhD, I just I used as I am a I consider myself more synthetic biologist, lesser geneticist, right? So during my PhD, I designed a novel dominant negative proteins. So if you know the transcription factor, and I like discuss with you about the HSF82, I again just want to give a focus on the transcription factor. Transcription factors are the key proteins which are basically taking the information from the surface to at the DNA level. So they are the n number of not the n, but they are the many type of the transcription factors. So as like the HSF82, there is a one class of the transcription factor is the basic leucine zipper transcription factor, right? So B-zips are basically a dimer proteins, right, which has a leucine zipper and a DNA binding region, like here, right? So what I did, so basically what happened, so in the wild type, the zip is basically binds to the upper stream of the DNA and it's participate in the transcription process. So what I did, I basically replaced the DNA binding domain with some like designer just new proteins. And what happens, and those proteins are basically in the acidic, rich in the acidic amino acids. So if you express those two proteins together, so what happens? So one of the monomer of protein is fused with another of the proteins, and this zip become closed. And now what happens? This zip close will not bind to its target site, and there will be no transcription, right? So I target the seed development phenomena. Right. So for that, so I expressed this protein into the Arabidopsis, and I found, there are like, I already published 2 or 3 papers, and you can Google it if you want to, like if you are interested in this study, that these are the, like most of the plants were not able to germinate the seeds, and the lines which have the seeds are very small compared to the wild type. That means these tools can be used for like to control the seedlessness. Now the question is that, okay, this is in the Arabidopsis, but we just want to eat fruits. So can similar things can be happen in the fruits? For that, so I use the CRISPR-Cas9 system in the UNC, and just try to like just generate the seedless tomatoes. And for that, I just target one of the complex PRC2. It is also the share the similar binding sites as like the ARP6, like the S3 lysine 9 methylation group. And when I just start knock out one of this PRC2 complex, the PRC2, I found that some of the fruits, like the tomatoes, have the seedless fruit phenotype. Means if you compare, this is the wild type, which has a lot of seeds, but others have no seeds. That means yes, it is possible. Right, so this is one of the few of the study, but at the end I just want to thank my mentors at the UNC, Greg, Jason, my PhD advisor Vikas, and late Professor Vinay Gupta at University of Delhi. Our collaborators, Naomi, Wolfgang, and Jonathan at the BSF, and our funders, the BSF, the NSF USA, and Department of Biotechnology for my PhD fellowship. Yeah, thank you. I'm open to questions.
Open for the question. Audience, it's your turn. Anyone interested to ask any query? Any doubt?
Well, sir, you can. The question is, so can we able to see the fluorescence in the root of the plants? For this thing, because the genes are expressing on the post-meiotic promoter, not in the ubiquitous promoters like the CMV or others. So for these, no, but for the others, yes, using different promoters you can see in the root, shoot, or wherever you want.
Right. Very interesting talk. I am from the Philwig lab. You might, Philip Wigge.
Okay.
The paper you showed S. Kumar,
Okay.
S. Vinod Kumar. So we proved it wrong, by the way. So that theory doesn't hold true, that ARP6 is temperature, high temperature stress. It is basically the phenotype is light induced, no more the temperature. And ARP6 is not sensing high temperature or high heat stress. It behaves that at 17 degree, by the way. So that is normal temperature. So that's why I asked you what's the temperature, flowering phenotypes, because most of the time temperature stress is leading to early flowering. That is the classic example of ARP6. And that is mediated through light and evening complex. But nonetheless, I am not quite sure how you say that these compounds generate the similar effect as heat. I am not able to understand.
No, like maybe I have not disclosed some of the identity of the chemicals, but some of the chemicals are very well reported, right? If you spray those chemicals and if you see, so how you will consider that the effects are similar to the heat stress? By looking at the phenotype or by looking at the expression of the genes in the terms of the transcription factors or maybe others. So when the people have grown the plants on those chemicals, they found the effects are similar as you give. So they did some heat treatment and they grow the plants on these chemicals and the effects were similar.
What are these effects? That's what I am wondering.
What?
Which effects are similar?
Like the expression of the genes, like the HSFA2 expression of the heat
That's
stress.
not the heat stress marker.
No, that's
HSF.
not. No, but they are the other phenotypes. So like the other physiological phenotypes.
That's what I was asking.
Yeah.
Physiological phenotypes of heat stress are few, longer hypocotyle, early flowering, etc., etc. And if you want to go on the gene, then one established heat stress gene is HSP70, which goes linear with respect to 17, 18, 19, 20. You can really, we call it plant thermometer. So you can see that if it is 22 cycles, plant must be at this temperature. But HSF A2 is a transcription factor, right? it doesn't change that much. And if you see, we published it in Cell Reports in 2018. We even used the null mutant of HSFs. And it has very different phenotype. So that's where I am a bit confused that how do you say it is similar? I don't see any similarity in your slides.
No, so for here, I'm saying the similarity in the terms of the mode of action, not the abiotic stress, but in the terms of the recombination.
Right. Let's come back there.
Yeah.
So I'm literally confused. So how do you measure the crossing over?
So yeah, so this is one of the like model, right? So in the meiosis, right, there are the 4 strands, right? So the mutant that we are using, the quartet mutant, right? So, and which is in the heterozygous state, not homozygous. Like 2 of the strands are expressing all these 3 markers and 2 are not, right? If there is a, if there is a recombination happening, means there is a breakage of the DNA, then you will see the 2 as a wild type, or maybe all and 2 as a recombinant type, right? And if it is NPD, then maybe all 4 are different. So by scoring all these individual tetrads on the pollens, we came to know that there is a recombination is happening.
So on that note,
Yeah.
if you are using this ARB6, if you to put them at even at 17 degree, in your own words, it will say the same thing. So where is the temperature? So it will nullify your effect, because ARB6 is the mutant which you are using. If you grow it at 17, it will show the similar. And Ian Henderson is actually published this last year in Nature.
ARP6 mutant?
Yeah.
So it will have the high recombinations?
Yeah.
Well, Ms. Ian Henderson, what paper I followed, that was published in, I think in 2000, it was in Science, in Nature Genetics. It was in 2014, right? So,
Recently.
Recently about the ARP6, that's more, I don't think so.
So let's not. So what I am saying that even at low temperature, ARP6 does show the higher crossover. So it doesn't get affected because of temperature.
No, what I'm saying, it's not about the low temperature, it's more about the high temperature. That the higher, it is not about, it is more about the temperature sensing. Right. I'm sorry, I'm not getting the question. No, it's fine. It's more about the effect.
More queries from the students? Anyone? Any doubt?
Actually, students of personalized medicine, right? So I was taking their class. So we do talk a lot about single nucleotide polymorphism, and some of them might be talking about, as you saw, recombination, crossover. I hope some of you should have understood, right? किसी को कुछ पूछना है?
It's fine.
सबके मार्क्स गए. आज का अटेंडेंस एब्सेंट. पूछ लो, नहीं समझ में आ रहा है तो पूछो. मैं कुछ सुना ही नहीं.
Thank you, Dr. Jain. It was a nice lecture, and hopefully our students have learned a lot from this lecture. So I thank you all for listening patiently this lecture, and I thank you, our director, sir, for giving this opportunity to me to organize this lecture. Thank you all students, all the faculty colleagues. Thank you very much. Now I request our Deputy Director, Dr. Amit Khadakwal sir, to felicitate Dr. Prateek Jain. Dr. Jain. Thank you all. Thank you very much.